Is it okay to sit here?
Yeah, perfect. Are you ready?
I think so.
Okay. Let's start from the beginning, where it all started.
Well, growing up, I never felt like I fully fit in with my classmates. I was smaller than the rest of them, I was awkward, and I didn't have many friends. I felt when I turned 13, everything would change, but I was wrong.
Why did you think everything would change when you turned 13?
Our 13th birthday was a big deal in my colony. It was the day our lights would turn on. On my 13th birthday, I woke up with a new excitement. I thought, today is going to be the day to change everything. But as the day went on, my concern grew. Eventually, it was 2 p.m. and my lights still had to turn on. Most of the time, they turn on by 12. My parents kept reassuring me that I was just a late bloomer and I was gonna glow, I just needed patience. Well, my 13th birthday came and went and my lights never turned on. It was heartbreaking and I needed answers.
And did you find them? Answers?
After a lot of hard work, I did. Before I get started, I have to check. Have you heard with mutation?
Yes, I have. But just in case others do not, could you please expand on the topic? So everyone is on the same page.
Yes, of course. Well, a mutation is essentially change in one's DNA. So when my lights didn't turn on, my family automatically assumed the worst. Something had to be wrong with my LUC gene. The LUC gene creates a luciferase protein which produces light for us fireflies. If something was wrong with my LUC gene, that would explain why I wasn't glowing.
Was that surprising to your family, or had this happened in your family before?
We were all definitely surprised because it has been unheard of for anyone in our colony, let alone in my family.
So, how did this play out when you went to school?
School was pretty rough, but I got through it. There were plenty of jokes and comments about my lack of light, but while other kids my age found it humorous, I found it curious. All I wanted to know was why. Why was I different? So I dedicated my life to studying the science behind my mutation, and here I am today to share it. I know what it's like to feel different due to a mutation, but I want all the young fireflies out there who might be struggling to know that it is okay. It's purely a scientific occurrence, nothing to be ashamed of.
You said you dedicated your life to studying your mutation, correct?
Yes, that's correct. Right after I finished school, I left my colony and began exploring all around the world. I studied in labs, talked to experts. I even found people like me share their experiences and knowledge.
And what did you find?
Answers. I finally found out why I am the way that I am. And actually, it's quite simple. It just has to do with what happens during this process called protein synthesis.
Protein synthesis? Now that is a term I'm not completely familiar with. Could you please elaborate?
Of course. Protein synthesis is basically the process in which cells make proteins, which occurs in two stages, transcription and translation. To explain it, I want to bring in my good friend Benedict. Growing up, I didn't get along with my fellow fireflies, so I resorted to finding comfort in my neighboring colony, the beehive. They never once judged me for not glowing, yet they understood me and treated me as their own. Benedict and I quickly became best of friends, and he too spent the better part of his life studying mutations.
Hi, thank you so much for having me. I'm so excited to be here today.
Yes, of course. Thank you so much for taking time out of your busy schedule to meet with us.
Of course, I'm very excited to share my learnings. Larry has been a very good friend of mine, and to be honest, his story is quite fascinating to me. My favorite way to explain protein synthesis is through my own life. As you know, bees like me love to make sweet honey. Well, protein synthesis is honestly not that different from making honey. I know that might sound silly, but it's true.
Wait, I'm lost. What does honey have to do with protein synthesis?
Well, they actually share a similar process if you think about it. To explain it better, I'm going to take you along with me for the day. Well, my day starts off by visiting the nucleus, aka the queen bee. I have to see her every day because the queen bee has instructions on how to make honey, similar to how the nucleus holds the instructions in the process of creating protein. I come from a family of copier bees. We take the instructions from the queen bee and transcribe them into a copy of the instructions, also known as mRNA. My family acts as that RNA polymerase of bees. We copy the instructions down. And just like that, you've learned the process of transcription. We've taken the instructions from the queen bee and transcribed them into a copy to take with us on our journey. Let's review. This is a strand of DNA. Helicase is used to unwind the double helix formation, and the RNA polymerase reads the DNA strand. It uses one of the DNA strands as the parent strand, or as a template to build the complementary strand of mRNA. A complementary strand means that the bases are of opposite composition and it follows the parent base rule. Each base has an RNA base it pairs to. Adenine on a DNA strand matches with the uracil on an RNA strand. Thymine, which is only present in the DNA strand, matches with adenine present on the RNA. When there's a cytosine on a DNA, it matches with a guanine and a vice versa. Basically, it creates a copy of DNA because DNA cannot leave the nucleus without getting destroyed. But that's just transcription. I haven't even made the honey yet.
Whoa. That is wild. I'm going to be honest. I thought you were going crazy at first, comparing honey to something I could never understand, like making protein for your body. But I totally see the connection now. But you said that's just transcription, which leaves what? Translation?
Exactly. Making the actual honey is very similar to the translation step of protein synthesis. Now that we have created our strand of mRNA, the complementary strand of DNA, we are able to translate it into a protein. In my case, honey is my protein I'm working towards. The copy instructions, which I got from a queen bee, or also known as the mRNA, must go to the site where the honey is actually made, the hive. The hive is representative of the ribosome and the site of protein synthesis. Essentially, when I go back to my hive to create my delicious honey, it's the same as the mRNA going to the ribosome to create proteins for your body. When I go back to my hive, I have all the instructions I need, and the bees all around me who work to make the honey will be known as the tRNA in the case of translation. They work together with both the mRNA and the ribosomes to read the mRNA and translate it into a protein. As you can see by looking at the diagram, there is something called a codon, which is a sequence of the 3 nucleotides in the mRNA, and they code for specific amino acids. Anticodons are seen opposite of the codons, and they are a type of RNA that carries amino acids to the ribosome during the translation step. Now, I've been talking about amino acids for so long, but what are amino acids? Amino acids are the building blocks of proteins. There are 20 different amino acids that can be combined in various sequences in order to create many proteins. Peptide bonds are what hold the structure of proteins, and they are formed when one amino acid reacts with another. The process begins when ribosome, subunit, and mRNA and tRNA all bind together. The process ends when a stop codon is reached and the newly synthesized protein is released.
That's right. In my case, because of my mutation in the LUC gene, the instructions encoded in my DNA got messed up. So imagine if the queen bee gave the copier bees the wrong instructions, resulting in a flawed copy. Similarly, my mutation led to errors or altering in the sequence of my bases in the LUC gene, which affected the function of my luciferase gene, protein.
Exactly, and just like how the quality of honey might be compromised if the instructions are incorrect or incomplete, the function of the luciferase protein may be compromised or non-existent because of the mutation. So even though the ribosome follows the mRNA's instructions, the final result may not be what it's supposed to be.
So basically the mutation is like a miscommunication or error in the instructions for making the protein, which results in the lack of your ability to glow?
Exactly. It's like having a mistake in the recipe for glowing, which causes the final product to be flawed. Now, my mutation specifically is called a missense mutation. There are many different mutations. Everything in the world has mutations, thousands of them. Some of them just happen to be called a silent mutation, which means it does not affect an amino acid and practically goes unnoticed. Without mutations, we would look exactly the same. Wouldn't that be boring?
Yeah, you're right. That would be boring. No one would be unique and have their own characteristics.
Yeah, exactly. Being able to understand this process helped me come to terms with my mutation and realize that it's just natural and not something I need to be ashamed of.
I know you talked about silent mutations, but you said yours was something called a missense mutation? What does that mean?
Well, going back to what Benedict was talking about with his experience of making honey, he talked about how the queen bee had instructions on how to make honey, and he, as a copier bee, or RNA polymerase, copied the instructions. Well, in the case of missense mutations like mine, we would have to look closer at the nitrogenous bases, or as Benedict would say, the specific steps the queen had. Say if the queen bee had instructions and they may not have been far off from what they were supposed to be, but one single step was wrong. It would affect the outcome of that step. In my case, one single nitrogenous base was incorrect in my DNA, which affected how it was transcribed into mRNA. Because the DNA and mRNA are now incorrect, one of my nitrogenous bases was incorrect. This resulted in a defective blow gene, and it's really interesting and goes to show how it is completely out of control for anyone. There's nothing wrong with me, I'm just very interesting and full of science.
That's very powerful. It's obvious that your journey was one for self-discovery and acceptance. How do you hope your story will impact other fireflies or animals struggling with similar challenges?
I hope that sharing my story and the science behind it, I can inspire others to embrace their uniqueness and not let their differences define them. It's important for all of us to understand that mutations are part of life and they do not take away our worth or value. By educating ourselves and others on this matter, we can create a more inclusive and accepting society for everyone.
Thank you. Benedict, for your help on learning the process behind the mutation today, and Larry for sharing your story with us. It's truly been enlightening.